Hello, I am Jeong. Today I want to talk about hemoglobin, the protein responsible for transporting oxygen in our bodies. Hemoglobin picks up oxygen molecules in the lungs and releases that oxygen to tissues where it is needed. In other words, it acts as a specialized carrier that can efficiently transport oxygen. To understand it simply, hemoglobin works like a delivery truck. Just as a delivery truck transports packages to various destinations, hemoglobin picks up oxygen from the lungs and delivers it to the tissues in our body. However, just like a delivery truck might face some obstacles on the road that could make its delivery difficult, hemoglobin also has several challenges during the oxygen transport processes. But because of its unique design, it can overcome these obstacles. Now I'm going to talk about 5 problems and see how hemoglobin solves them. Problem 1. Oxygen is poorly soluble in water, making it difficult to transport enough of it in the blood. How can we ensure that hemoglobin effectively captures oxygen molecules? Hemoglobin is uniquely structured to effectively transport and store oxygen. It consists of 4 subunits, each containing a heme group for oxygen binding. The heme group is a planar structure with an iron ion at its center. This structure creates an optimal environment for binding oxygen. The iron ion can exist in the Fe2+ state, allowing each heme group to reversibly bind and capture oxygen. Also, hemoglobin can cooperatively bind. When the first oxygen molecule binds, it increases the affinity for the remaining heme groups, and it makes it easier for other oxygen molecules to bind. This unique design and cooperative action make hemoglobin to efficiently capture and transport oxygen molecules. Problem 2. Free iron can form highly reactive oxygen species that damage DNA and other macromolecules. How can we safely store and release oxygen? In hemoglobin, the iron ion is not free. It's embedded in a heme group, which stabilizes the binding. And the heme group is surrounded by specific amino acid residues, particularly histidine. Histidine residues form coordination bonds with the iron atom and make the iron remains in the Fe2+ state. Also, there is an allosteric regulation. When oxygen binds to hemoglobin, T state is shifted to the R state. This facilitates oxygen transport and prevents the oxidation of Fe2+. Problem 3. Small molecules like carbon monoxide and nitric oxide have a much higher affinity for heme than oxygen. How can we reduce the binding affinity for CO while maintaining a strong affinity for O2? Hemoglobin enhances structural specificity to reduce the binding affinity for CO while maintaining O2 affinity. O2 binds with a directionality to the iron within the heme group. On the other hand, CO binds perpendicular to the plane of the heme. So the modification of the hemoglobin structure can stabilize O2 binding and hinder CO binding. And also, hemoglobin is an allosteric protein that undergoes conformational changes based on O2 binding and release. When O2 binds, it transitions to the R state, which promotes the binding of additional O2. In addition, competitive inhibitors that are similar with O2 can prevent CO from binding. Lastly, hemoglobin dynamically regulates its binding properties in response to changes in O2 concentration in the body. Through these mechanisms, we can reduce the binding affinity for CO while maintaining a strong affinity for O2. Problem 4. We need to quickly absorb oxygen in the lungs and release it in tissues. How can we perform both functions effectively? Through cooperative binding, hemoglobin can regulate the binding affinity for O2. When one O2 molecule binds to a heme group in one of the subunits, it induces a conformational change that increases the affinity of the remaining subunits for O2. This means that hemoglobin can quickly bind O2 molecules in the lungs where O2 levels are high. It ensures maximum oxygen uptake. In contrast, in the tissues where O2 levels are lower, hemoglobin can efficiently release O2. Also, hemoglobin is regulated by various allosteric effectors. Through these effectors, the production of CO2 and H+ lowers the pH. So it stabilizes the T state of hemoglobin, reducing its affinity for O2 and facilitating O2 release. Problem 5. In high altitude conditions, lower atmospheric pressure reduces the concentration difference for oxygen exchange. How can we maintain a reasonable oxygen exchange rate under these conditions? In low oxygen environments like high altitudes, hemoglobin adapts structurally and increases its affinity for O2. These adaptations allow hemoglobin to absorb O2 more effectively even when the concentration gradient decreases. Also, there are BPG, the oxygen carriers. By adjusting the concentration of BPG in the blood, the affinity of hemoglobin for O2 can be altered. At high altitudes, increased levels of BPG reduce hemoglobin's O2 binding, facilitating more efficient release of O2 in tissues. Lastly, when the oxygen level is low, our body increases the production of red blood cells. Then oxygen carrying capacity is enhanced, so sufficient O2 can be transported even in high altitude conditions. This process is primarily regulated by erythropoietin. These mechanisms maintain effective O2 exchange in low pressure environments like high altitudes. I explained the challenges hemoglobin faces while efficiently transporting oxygen and how it overcomes. Because of its unique structure and regulatory mechanisms, hemoglobin can supply our bodies with oxygen even in various environments. I hope this video is helpful. Thank you for watching.